So if you're on a long cross-country run of 10 hours or so, you probably, unless you take additional fuel along the way with you, high carbohydrate form, the goo that my husband uses when he's cycling, you're going to start to break down lipids, all right, to provide, or amino acids to provide additional fuel. But this, as I indicated, this can go on for hours and hours as long as you have oxygen and some fuel source available. Now, let's look at the turkey muscle, okay? This is to prove to your family and friends how much you know on Turkey Day. So there are two components, and these used to be the two major ones, and they found three, and now each of these is broken down into subsets, but we're going to limit to these three major components. So we have slow twitch, which remember I said had to do with the speed at which the myosin head breaks down ATP. And we have fast twitch. We have two different kinds of fast twitch. So again, the speed has to do with the ability of the myosin ATPase. So this is a slow acting myosin ATPase. And this is a fast acting myosin ATPase, and this is also fast acting. Now, the next, and this is really what gives the coloring to the muscle, has to do with the amount of oxygen available and whether they're using the aerobic or their anaerobic component. So we have slow twitch oxidative fibers, meaning they're capable of using aerobic metabolism for extended period of time. So what organelle do we use to make ATP? Mitochondria. So these have high numbers of mitochondria. We need oxygen for the mitochondria to utilize in the aerobic metabolism. So we have a good oxygen supply with a high number of capillaries. And within the muscle cell, we're able to store a limited amount of oxygen on a protein similar to what we transported in red blood cells. That's called hemoglobin. This is myoglobin. And it's primarily the presence of the myoglobin that gives the dark color to the meat. All right? So then we have fast twitch, which are glycolytic. Meaning they use it primarily through glycolysis, which is not aerobic. So they have the opposite of all of these. They have a decreased number of mitochondria, fewer capillaries, and little, not small, but small amounts. Hemoglobin. This would be the white meat. And then we have fast twitch that is also oxidative. So if we take all of these characteristics, you would see them with these as well. Right? So what do you think? Yes?
That's supposed to be myoglobin, not hemoglobin.
Thank you. So what do you think the kind of muscle cells in a hummingbird's wing would be?
Fast twitch oxidative.
Fast twitch, not oxidative, because they don't do it for a very long period of time. Maybe a migrating bird, okay, a migrating goose or duck is going to be flying all day. They would be fast twitch oxidative. But hummingbird is really fast, but it doesn't do it for more than a few minutes at a time. It does this and then go over and rest, and it does it, maybe hovers in place for a little while, but it doesn't do it for hours. Okay? So hummingbird's wings would be fast twitch glycolytic. That's kind of what I can't fly at all. I can't do it fast enough. I can do this for maybe 15 minutes before I start to get exhausted. Okay? Maybe a little bit longer than that, but I can't do this for 10 hours. All right? So, fast twitch, somewhere in between oxidative and glycolytic. What about the muscles in my back? Slow twitch, but I can do it forever. So that would be the slow twitch oxidative. All right? So my back doesn't get tired. Well, I used to date this history professor, and he came up to Maryland to stay with our family for Christmas, and he wanted to go visit all the museums in Washington, D.C. He had to read every little card under every little plate and cup and saucer that any first lady had at the White House. And I can walk 30 miles, but I can't stand and stand and stand. We obviously never got married. And I was just like, let me just walk. I could walk circles around the entire museum 5 times while you're reading that one little card. My back just gave out. So, but generally in humans, our muscles may be primarily one type or the other, but in any given muscle, it's going to be a mixture. Okay? So as I said, a sprinter is going to have predominantly the fast twitch glycolytic. The fastest runner in the world in a marathon would have more of the fast twitch oxidative. Okay? But it's a predominance of one, not completely one or the other. You can't change these by deciding you're going to be, when my husband has kind of, well, not that big, but they kind of tree trunk thighs. He calls them, not me, he calls them. When we first started dating, he asked if I thought he was good looking. And I said borderline.
Ouch.
We've been married 23 years now. And I can't say, honey, you look really good tonight, or I really think you're handsome. And he looks at me and he kind of grins and goes, borderline maybe. I said, can't you improve with age? I think you're past the borderline stage now. All right, so he decided to run the marathon. When he turned 50, it's like, okay, I'm going to run the California International Marathon. So he trained for 5 months. His whole physique changed. But it was more that he developed the fast twitch oxidative fibers, that he converted the fast twitch glycolytic to fast twitch oxidative. That's genetically determined, interestingly enough, by the type of neurons that innervate them. That seems to be the factor that determines whether it's glycolytic or oxidative. Okay? So we do, but we don't see the distinctive, you don't have white meat and dark meat in humans to the extent that we see it in animals.
To recap on that, you said that you can change.
You can increase the size of the ones that are what are a particular type.
All right.
And we do that by adding actin and myosin. That's how we increase muscle size.
Okay.
All right, so if you use those more,
No, I'm under the impression,
Yeah.
or I was under the impression that you can work with what you were given,
Right.
period.
and you can enhance it by increasing the size, but you can't change.
Right.
Okay, so if you focus on long distance running,
You'll get better at it, but you're
you
not
get
gonna
better at it, but
change-
you're not going to start changing muscle type. Exactly. So Bolt is not. Have you ever heard the theory that people tend to become what their names are?
Lightning bolt.
So who's the long distance?
Usain Bolt.
Yeah. Okay.
Sprinter.
And people who work as a florist are called flower or peony or something like that. There's almost a subconscious. And yeah, you can find examples of anything, but yeah. out of context there. All right, so just a few minutes on the difference between smooth muscle and cardiac muscle, and we'll be ready for muscle identification next week.
So never knew anxious. Why explains all the onions?
Cardiac muscle.
Explains a lot.
So this starts on page 38 with smooth and page 40 with cardiac. So skeletal muscle is a cylinder, the length of the muscle bundle. It has multiple nuclei just under the cytoplasm. And it demonstrates the A and I banding. So we see these striations. Innervation is through general somatic efferent or voluntary neurons. Okay, we have all the actin, myosin, sarcomere kind of components we've gone through. Smooth muscle cells are shorter, so this actually goes on and on and on. I don't want to give you the impression that they're the same length. And they are what we call fusiform shape. So these are cylindrical in shape. These are fusiform, meaning they have tapered ends. They have a single nucleus, which is kind of a rod shape, elongated, found in the center of the cell. And it does not have banding. All right, so sarcomeres are present here. No sarcomeres here. It definitely has actin and myosin, but more in a kind of, have you ever seen those baskets kind of formed by tying knots? And you have the appearance of a reticulation, a network, kind of like that. So the actin and myosin are overlapping, but not in an organized fashion, so no striations. In the case of skeletal muscle, calcium ions bind to troponin. In the case of smooth muscle, calcium ions bind to a molecule known as calmodulin. We have a sarcoplasmic reticulum present in skeletal muscle. There is no sarcoplasmic reticulum present in smooth muscle, just vesicles of calcium ions. So calcium becomes important, but not in an organized fashion. So I've given you the physiology of smooth muscle contraction, but what I want you to know are the bullet points on the top part of page 38. Smooth muscle has gap junctions present between the cells. This fusiform shape allows the muscle cells to form a sheet. Yes?
What does that say? Calcium binds to?
Calmodulin. C-A-L-M-O-D-U-L-I-N. In between the muscle cells, we have gap junctions that allow them to send the depolarization from one cell to the next. So in a cross section, if I turn the light off here, you can see the top image, those are in cross section. See how the diameters are, we have small cross sections and large ones, and some with nuclei and many without. That's because as we cut through this cell, if we cut at the tapered end, we're not going to get a nucleus. All right, we have to cut right through the cell. So when if I make a section through like this, I'm going to see a cell with a nucleus, a cell like this. So I'm going to get different appearances depending on where I cut in that fusiform shape. Okay? And our innervation is general, visceral, efferent, or involuntary. This is our sympathetic and parasympathetic. I have an image of a smooth muscle cell contracting on your lecture notes, similar to what you see here. All right, so it kind of just makes a twist, like you're wringing out a washcloth. And you see the whole smooth muscle cell shortens and kind of twists both the cell and the nucleus. All right, then we get to cardiac muscle. Cardiac muscle is not shown very well up here, so I'll diagram it. Cardiac muscle has characteristics of both skeletal and smooth internally. Externally, it's a little bit longer than smooth, but not as long as skeletal, and it's branched. It has a single nucleus that's kind of shaped like a boxcar on a train, just a big old rectangle here. It does have sarcoplasmic reticulum and striations, so in that manner it's similar to skeletal muscle. So we see the actin and myosin in these A bands and I bands. So single nucleus. Sarcomere, sarcoplasmic reticulum, troponin, but it's a general visceral efferent neuron. If you recall, it's going to be autonomic. And like smooth muscle, it's attached via gap junctions. Okay. So at this site here, I'm going to draw this a little bit larger here. So here and here we would have gap junctions that allows the current to flow from one cell to the next. And then here and here, holding the cells together, we have desmosomes. So gap junctions, desmosomes, and this is seen, you have a picture of this in your lecture notes, it's better than on screen. These are much darker than the striations and are known as an intercalated disc. You can see the striations on page 40, but then there's these really dark lines, and that's where we have the overlapping membranes of two cells joining together. Those are known as intercalated discs. Okay. So we'll pick up with the peripheral nerve, and I'll have the rest of the lecture notes should be here on Monday. You want a break from finishing your take-home? A few of you that haven't finished yet. You might want to pick up a pack of 3x5 or 4x6 cards, because as we go through the skeletal muscles, it helps to make flashcards flashcards for these. You can print off black and white or colored pictures from online to put on one side and the information on the other. But more than just a memorization, more than just going through the cards as you're, waiting for water to boil or something. But you can, I have questions on the study objectives. Identify muscles which cross the shoulder joint anteriorly. Identify muscles that move the jaw. Identify muscles that are going to extend the thigh. Identify all the muscles that attach to the ischial tuberosity or to the mastoid process. And you can just kind of identify all the muscles innervated by the median nerve. And you can kind of go through the cards and just pull them out. Okay, so there's a great variety of ways to use that. I would also pick up the water-soluble coloring pens. Not permanent, not Sharpies, not the dry erase. It'll be blistered red by the time you try to, are they water soluble? Do they wash off? Okay. And color yourself, all right? So I will bring some to class on Tuesday and I'm going to draw muscles on my face. All right, so as you draw these muscles, is the camera on?
We can take pictures of that, right?
Yeah. You can have nude coloring sessions.
She was like nude coloring session. She did.
Who attends them, I'll leave up to you. I suggest this with blood vessels in Bio 431. I had a student, I actually give extra credit for that, in Bio 431. I had a student go to an adult store and buy a doll, blow-up doll. She left it with me, and after it was in my office for 2 or 3 weeks, stuff in half and down the paper sack. I really don't need this hanging around. I've had parents, pull T-shirts onto their kids and draw on those.
Wearing like muscle leggings.
Yeah. You can buy them already done. I thought about coming to class sometime as a surprise on Halloween for my sex day class, but just the process of drawing it. So this muscle comes off of the coracoid process and goes to the humerus. So I'm going to draw it like that. Okay, this muscle functions to flex and to abduct. So not only can you visualize it, but you can feel the pressure, the wetness of the pen as you draw it on there. All of that helps to create memory. Okay, as you're working with these muscles. So either doing it on yourself. You guys were telling me about drawing on the big mirror, so you can draw on that too. Whatever helps to visualize these components. Give a massage and identify the muscles that you're pressing on for that, okay? So all of those are various learning techniques. You don't want to just sit in a room and stare at a piece of paper and memorize it. I mean, that may be a place to start, but it's not going to stay with you very long. All right, why don't you guys, we will, let's see, start the quiz in 20 minutes. Okay? So take a break. We have the quiz and then we have the wet lab to do.